Okay, so our next speaker is Professor Mark Molloy, who is representing TCE6.
We've been in the colorectal TCE working to increase capacity, I guess you could say, for translational research to use various molecular technologies. To do this, we've had 3 projects in our TCE. First project is the Sydney 1000 colorectal study that was kicked off a number of years ago before I joined the group. By Connie Alexander, Kelly and Stephen Clark. And the idea of that study was to prospectively collect a variety of molecular specimens and information around people with colorectal cancer across their journey. In hindsight, this has proved to be very ambitious to do this. We've been able to collect about 71 patients at baseline, and about a third of those we've got some sort of follow-up collection of specimens as well. And we hope that we can continue on this study with a PhD student next year, which is really a key aspect, we think, in enabling this type of a project to be successful. Number 2 is looking at precancers. So we know that bowel cancers develop from pre-malignant growths, known as polyps. So we've got a prospective study underway where we collect polyps, we collect mucosal biopsies as well, blood and oral swabs to look at microbiome data as well. And that study's underway, and I'll show you some data from that in a minute. And Project 3 was a recognition that we didn't have a lot of great knowledge of our patients that were coming through the centre with rectal cancer. While we had plenty of specimens available in the pathology archives, we lacked linked clinical information to be able to utilise those for molecular research. And so Jen White, who joined us earlier this year, has now been able to assemble that into a database of over 400 patients. So we hope to be able to utilise that as a resource in the coming years. So I'm just going to talk very quickly about Project 2, the IMAP Bowel project study. So we've been working on this for about 12 months with Alexander Angle for patients undergoing colonoscopy here in the public hospital. We've enrolled 164 patients at the moment. We've collected 67 polyps and adjacent mucosal biopsies from those. Most people will give us plasma and oral swabs as well. And with those polyp biopsies, we've subjected those to whole exome sequencing to try and understand the features associated with those polyps. Unlike cancers, there's not a lot known about these pre-malignant lesions, so we think this is an interesting opportunity. And you'll probably be aware of the concept of the tumour mutational burden that happens in cancers, and can people use that as a risk factor. So we're applying a similar approach in these pre-malignant lesions to see whether the number of mutations can be used as a risk factor to help guide potential future screening intervals for repeat colonoscopies and other risks of the likelihood of conversion to cancers. So I haven't got time to go into this, and you probably can't see it, but other than just to show we've started collecting data, we've got whole genome sequencing there, and we do see a range of spread of the mutational burden in these lesions. Interesting, some of the big ones show a lot of mutations, as you might expect, but similarly, some of the big ones also show very few mutations, and so there's perhaps an opportunity to redefine risk associated with this. We've also been looking at the microbiome in the mucosal specimens. You know, most people do stool samples for microbiome analysis. We're using the actual mucosa specimens and comparing that to the oral swabs, because we know there is a connection in colorectal cancer with bacteria that are normally residing in the oral cavity being found in colorectal lesions. So we're comparing those things. And again, I won't have time to go into this, but just quickly on the right there, you can see that this is a relative abundance of reads for the bacteria Fusobacterium nucleatum species, which are known to be found in colorectal tumours. We can also see that in our patients with polyps that yes, we can find evidence for this bacteria phylum in those with polyps, but in people that have colonoscopy and have no polyps, we don't see any evidence of that type of bacteria. And so this is just an indication that we can hunt this data in much greater detail and see if there's any associations of particular enterotypes of bacteria with their clinical features. So in terms of success, we've had 3 students at various times working on these projects. We published our first paper from the Sydney 1000 cohort earlier this year. We've had some success, I guess, with funding. We've got Cancer Council funding. We're still in the running for a large translational program grant, which should be announced in the coming month, which would be fantastic if we could get that. It could give us funding for 5 years to continue this type of a study. And yeah, that's it. Thanks.